It's my pleasure to now introduce the second session of this symposium. We'll be starting with the topic of taming the tumor microenvironment ecosystem. And our first speaker will be Dr. Greg Delgoffe. Dr. Delgoffe comes to us from the University of Pittsburgh, where he is an assistant professor in the Department of Immunology.
Right. Cool. Can you all hear me? All right, we're good. All right. Thank you very much. I'm honored to be invited to this symposium. It's and I felt very welcomed since I arrived yesterday. So I'm going to talk to you today. We're going to switch gears. Not a whole lot of sexy imaging or anything else like that, but we're going to talk about something that is generally I think we're now becoming a little bit more generally appreciated, but the fact is it actually goes back a century or two. Understanding the role of metabolism both in the generation of an immunosuppressive tumor microenvironment and how those factors play with immune cells. These are my disclosures. The biggest one is that I'm a founder and scientific advisor of this company, TTMS. I won't be discussing anything really related to that work in this talk, but just in the interest of full transparency. So the reason why we're excited about this question is that we know that the process of engaging an immune response, and this is work that goes back a couple decades now, is a metabolically demanding process. So a naive T cell, right, these cells that sit and wait, right, they need to be able to persist potentially for a lifetime, having never seen their target antigen. So they have to be extremely quiescent. But then upon activation, need to enter into an extremely proliferative phase in order to generate a clonal army. Now, and so there's been a lot of excitement around the role of how does a T cell fuel that response. And what we now know is that as T cells go from naive to early effector cells to being able to infiltrate into a target tissue and then scan and destroy, that they have very, very different metabolic needs. But more importantly, after an immune response is over, a cell needs to be able to go back into that long-lived quiescent state, but ready to see antigen again. And so we now know that memory T cells are characterized by having a high degree of mitochondrial mass and this value that we call spare respiratory capacity, which is kind of like parking your mitochondria in reserve and keeping them in this healthy metabolically primed space. And so we've been really trying to understand how do T cells behave when they're stimulated in those dearth environments. And that's really how things come. So this is the one cool image that I get to show you today. And this is a tumor section where we are able to mark areas of high glucose uptake using a fluorescent glucose tracer. This is a 1-amino Cy5 glucose. Which is kind of, imagine it like FDG, but it's fluorescence. And this is a marker, an antibody stain for a marker called pimonidazole. It's a radiosensitizer. Old radiosensitizer doesn't work very well, but it does mark hypoxic regions very well. You can come back with an antibody and stain for it. The reason why I'm going into detail is because this marker, this pimonidazole marker, is going to come up again and again throughout the talk. So you can see in a tumor microenvironment, it's not this kind of like hypoxic core surrounded by healthy tissue like that we see in pictures, but rather there are zones of hypoxia, there are zones of high glucose uptake, and T cells are kind of smattered around there. There's an experience of all of these different metabolic parts. So we got excited about this because cancer is driven by metabolic deregulation. If you believe Otto von Warburg, he thought that was the reason that you have cancer cells. However, we do know that includes increased nutrient uptake and this kind of deranged glycolytic and oxidative metabolism, and that creates a landscape that's distinct from normal tissues. And so the tumor microenvironment that's driven by that metabolic derangement generates both a loss of essential nutrients, things like glucose and oxygen, but also a buildup of toxic byproducts. And so we wanted to really ask the question, is that in the patient population, is this a thing? Are all tumors turned up to 11? Or is it different from patient to patient. And so this is some published data. It's the only piece of published data that I'm going to talk about. The other two stories are actually are nascent works. But we hypothesized that we could get insight into the tumor microenvironment by metabolically profiling the tumor cells. And so one of the ways that we do this is using an instrument called a seahorse. This gives you a real-time view of the metabolic proclivities of cells. So how oxidative they are or how glycolytic they are. And these are the readouts that we're going to use in a kind of a two-dimensional plot. And so what we basically did at first, and we in Pittsburgh, we worked very heavily with the melanoma program, and so we started by just kind of thawing out all the human cell lines that we could find in the freezer, and we put them into the seahorse. In the same media, but asking them what kind of metabolism they perform. And we can get these two different readouts. And here are the data that you can see is that it's kind of all over the place. You have some cells that are truly hypermetabolic, like this A375 cell line that everybody loves. And you have other cell lines that are not so hypermetabolic. They may only deregulate one type of metabolism, like this M308 line is solely oxidative and does actually no appreciable glycolysis, that kind of fermentation of glucose into lactic acid. And you have other ones that do kind of the opposite, like this M255 line. And so there's a lot of heterogeneity in these cells that have been in culture for decades. And so we wanted to understand a little bit more about what kind of environment these tumors might produce. So we did what most tumor immunologists are loath to do, which is we put them into immunodeficient animals. And so what we do is we let those guys form a tumor in an NSG mouse, and then we infuse them with that p-manitazole molecule, that molecule that could mark hypoxia. And we asked, could we see a difference in the generation of hypoxia based on the tumor cell metabolism? And here's some of the data with that hypoxy probe antibody, that thing that this molecule I was telling you about before, is that if the cell was characterized by having a high OXPHOS signature, those cells, those tumors that formed at a threshold size, so all of these are the same size tumor, had produced different levels of hypoxia. But this guy down here, this M255 line, which does far less oxidative metabolism, didn't really produce a lot of hypoxia. So it seems kind of like, okay, the oxygen has to go somewhere, but we were trying to link the OXPHOS signature of the tumor cell with the generation of hypoxia in vivo. And we think we've done this here, and I'll show you some data in competent models that we think that we're making some strides here. So that's, of course, in cell lines. Could we do the same thing from patient samples? The beauty of this seahorse instrument is that you don't need the millions of cells that you used to have to put into a Clark electrode. You could do this from very small numbers of cells. And so we hypothesized we could do this from biopsies. And so all this is going to be from metastatic melanoma tumor biopsies. We have a sentinel lymph node project as well, but I'm not going to talk about that today. And so we have this IRB-approved immunometabolic profiling protocol where we take a section of the tumor, we do some embedding in Ducodex, we sort away the leukocytes. So all the CD45 positive TIL, we have a sorting panel, and then we do all kinds of cool stuff. And then we take the tumor cells and subject them to a metabolic profile, okay, direct ex vivo. We can do this with melanoma because they like plastic. We try to do this with other cancer models, and we can talk a little bit about that offline. So if we go back to the same kind of plot, OXPHOS versus glycolysis, what we found is the same heterogeneity in the patient population that we observed in the cell lines. That some folks were characterized by hypermetabolic tumors, while others had solely oxidative or solely glycolytic tumors. And so I'm putting this up because as an immunologist, I like to subset things, and so we started subsetting these patients into these groups based on how energetic or singly metabolically active their tumors were, and then, because of course this was patient samples, we could then profile the matched PBL and TIL that we got from each of these individual patients. And so when we do that kind of work, we create, we're able to get a sense of linking the tumor cell metabolism to the T cell metabolism, but not in the same environment. Remember, we've sorted away those T cells. So in order to account for any differences in T cell receptor expression and things like that, we just did a very brief chemical restimulation to ask what kind of cytokines could these T cells make if they were given a chemical activation. And if you look at T cells that we isolated from those very energetic tumors, and we, again, re-stimulated them in isolation, what you can see is kind of classic tumor-induced immune suppression, is that PBL from these patients were competent to make TNF and interferon gamma, but the T cells from the tumor were far less competent to do so. And again, if we look at T cells that came from those more indolent tumors, those tumors that didn't really deregulate either their OXPHOS signature or their glycolysis signature, those T cells look just fine. They really were no different from the circulating T cells from the same patients. But when we started looking at those individuals, that's when things got really, really interesting. Is that if T cells came from these hyperglycolytic tumors, they weren't that bad, especially when they were isolated away from the tumor cell and restimulated, is that their fate hadn't really changed. And in fact, they were, there was statistically no difference between the glycolytic tumors and the quiescent tumors. But it was really coming from this hyper-OXPHOS signature that rendered the T cells less polyfunctional. Being in that highly oxidative environment changed the fate of those cells such that they were less functional even when they're no longer in that environment. And so, of course, every one of those patients is individual with their own immune system, their own tumor. So could we assess the individual contributions of these metabolic pathways in a more controlled system? Could we reverse translate it? And so we used Marcus's mice. And instead, the original plan was to make single-cell clones and that there was going to be metabolic heterogeneity of individual clones, and it was going to be this really cool experiment. And we ended up wasting a lot of time because by the time you took the single cells out and you made clones, everything was turned up to 11. The process of the in vitro culture wouldn't give us the heterogeneity that we really wanted. So instead what we did is we took a single clone that we made from a melanoma that formed on this animal, and which we call clone 24, and this clone was extremely energetic, completely insensitive to immunotherapy, and very, very, very aggressive, but instead then targeted a single metabolic pathway in those cells. And we chose two obvious ones. One was the glucose transporter, SLC2A1, to prevent that glycolytic metabolism, and another one was this molecule called NDUFS4, which is a nuclear-encoded component of mitochondrial complex 1, that first step in the electron transport chain that drives oxidative metabolism, all the things that you tried to forget about from undergraduate biochemistry. I'm sorry, it's all going to come back to haunt you now. And here's what happens. If you take the control cells, they're highly, highly energetic. If you knock down the glucose transporter, you then recreate the solely oxidative tumor cell line. And if you knock down that mitochondrial complex 1, you create solely glycolytic tumor cells, okay? This is all RNA interference. We used CRISPR as well, but there's some caveats which we can discuss offline. But now, these are all derived from the same single cell. So now we can put them into tumors, or into animals. And that's what we do. If we take these tumors, interestingly enough, remember, this is in a very aggressive cell line. And so it doesn't matter. The cells always form tumors, which was a little bit of a surprise to us. But in the absence of any immune stimulation, the cells all form tumors at the same rate. But it was when you looked inside the tumor that things changed. And the big shift that we saw is that if you knocked out the glycolytic metabolism of the tumor, nothing changed. Nothing immunologic changed. The cells were, that the state of the T cells that were inside was similarly poor. However, if you inhibited the oxidative metabolism of the tumor cells, lowered that OXPHOS signature, the T cells had a more favorable profile. We had less terminally exhausted T cells, the cells that are PD-1 high and expressing multiple co-inhibitory molecules. And we had more of the PD-1 intermediate cells, those stem-like cells that are thought to be the true targets of PD-1 blockade. If you don't like this kind of nomenclature, that's fine. We also just restimulated them and showed that if T cells came from this less oxidative tumor, they were more competent to make cytokines when sorted away and restimulated. So this is interesting, I guess, looking at kind of subtle shifts in the tumor microenvironment. But what does the tumor microenvironment actually look like? And here's that same hypoxia imaging that I was talking about before. These are whole tumors stitched together. You can see that when you knock out that oxidative metabolism, you truly knock out hypoxia. If you knock out the glucose metabolism, you actually drive hypoxia, which is a very important point. So when you knock out one fuel source, the tumor cell has to use the other. And so what you can find is that these are not staining artifacts. These are actually depots of T cells that are stuck on the outside of these hypoxic regions. But when you lack that hypoxia staining, the T cells are able to penetrate deeper into the tumor bed. And far more importantly, if you take these mice and then put them on immunotherapy, This is where the key comes out, is that the glycolytic metabolism, if you knock it out, doesn't make any bit of difference. Those T cells, those tumors do not respond to anti-PD-1. However, if you knock out the oxidative pathway, you then start to see responses to PD-1 blockade. So this is pretty exciting. This linked the oxidative metabolism of the tumor with the inability to respond to checkpoint blockade. And we're not the first people to talk about this. In fact, RNA sequencing of tumors that have innate resistance to immunotherapy, the number 2 gene ontology list is hypoxia. So we know this, but it was nice to confirm this in a tumor cell intrinsic way. So now we went back to the patient samples and we asked the question, could we then do our oxphos versus glycolysis signature and ask some questions about whether or not those patients are going to respond to immune-based therapy. So this is all assessment of that tumor cell metabolism, but this is in a cohort of patients, all metastatic melanoma, about to go on either Nivo or Pembro monotherapy, and this is all pre-treatment biopsies. And if you then look at this, if you look at this bivariate plots, and you color it based on who goes on to progress, you can see that if the tumor cell has deregulated oxphos signature, it is, that patient is more likely to progress on anti-PD-1. Here are the data broken down by non-responder versus responder, and here's the overall survival data, whether or not the cell is an oxphos high signature or an oxphos low signature. Even in patients that respond, the duration of response we could find also would stratify with the oxphos signature. And we've seen some similar data to this being shown with a couple different groups, namely Mike Davies' group at MD Anderson, as well as some work from Mike Curran, also in Houston. So we think this is an important idea of understanding what the tumor cell metabolism is and how does that relate to immunotherapy response. Now, that's a lot of work. Taking these digestions, getting out the tumor cells, putting them into the seahorse, it's exciting from an academic perspective, but the fact of the matter is, could we just look for hypoxia? And that's one of the things that we did. We used we are now treating people with pimonidazole. It has an IND, it's a safe tracer that you can use. However, you need to pulse patients, so you can't look retrospectively. But we have looked at hypoxia by a pretty standard immunohistochemistry antibody for carbonic anhydrase 9. It's a target of hypoxic signaling. It's not true hypoxia, but it's close. And this is if we get strong membranous staining for CA9, we're able to elucidate the PD-1 non-responding patients in a pre-treatment biopsy. So now I'm going to transition into 2 pieces of unpublished work that I think this group might appreciate, really kind of digging into the immunobiology of some of the deleterious populations that are present in the tumor. So what's so bad about hypoxia? We talk about it a lot. And in fact, if you've been in the news, the Nobel Prize, of course, was awarded for hypoxia this week. But oxygen tension has this very interesting relationship with the immune system, in that if you take T cells and you just grow them up in the presence of hypoxia, they actually become great T cells. They're very good T cells. They kill targets very well. But we know that hypoxia has been shown to also have inhibitory pathways. So I actually put this up here. Hypoxia has been shown to both inhibit, stimulate, and have no effect on T cell function if you look at the right papers. So sometimes it's with genetic systems, knocking out HIF1 alpha, knocking out VHL. Sometimes it's culturing cells under low oxygen tension or no oxygen tension, but it's a little bit unclear. But the important thing is that hypoxia in the tumor microenvironment is a little bit different. It's not just the loss of oxygen, but then the presence of deleterious byproducts, right? So as oxygen tension drops, you don't just lose oxygen, you gain something bad, which is the production of reactive oxygen species. That's the first thing I'm going to talk about. And as the cell becomes hypoxic, it also becomes hungry for glucose. And as you break down and ferment glucose, you lose some glucose, and that's important. But we also build up lactic acid. So those 2 things are the next things I'm going to talk about: hypoxia and acidosis in the tumor. So we hypothesize hypoxia itself may not be a direct signal, but it changes the way that signals are received. So I'm going to go back to some original data, understanding what happens to T cells when they enter the tumor. And what we found is that intratumoral T cells a few years ago have very, very striking metabolic defects. This is in melanoma, B16 melanoma-bearing mice, and we're able to profile using a fluorescent glucose tracer, akin to the one that I talked about before, this one called 2-NBDG. and mitochondrial mass with this dye called mitotracker, you could find that T cells that infiltrate tumors have very, very striking metabolic defects, an inability to take up this tracer, and a loss of functional mitochondrial mass. This occurred in many solid tumor models. We also saw it in human patient samples, which you can see up here. This is in melanoma patients, but we observed the same thing in head and neck cancer, ovarian, and lung, is that when you look at a patient's PBL versus the tumor-infiltrating T cells, we can see this mitochondrial atrophy, that T cells lose their functional mitochondrial mass. And that is actually also predictive of response to immune-based therapies. So we linked this loss of functional mitochondria with the ability of these T cells to persist and produce cytokine. And what we found, just again, I'm making a very, very long story short so we can focus on the cool new stuff, is that as T cells terminally differentiate to exhaustion, and that's a hot button word, but I'm going to keep using it, this occurred due to downregulation of this molecule called PGC1 alpha. It's the PPAR gamma coactivator 1A, if you want to look it up. It is a transcriptional coactivator that programs a process called mitochondrial biogenesis. And as T cells differentiate from being PD-1 low, intermediate, high, and then becoming terminally differentiated, they repress the expression of this molecule. Okay? And that was the smoking gun for why these mitochondria failed and that T cells lost this functional mitochondrial mass. And so this has been very vexing to us. And my graduate student, what she's now, she just got her PhD, but she wouldn't let it go. She didn't know whether or not T cells became exhausted and that made them metabolically insufficient, or was metabolic insufficiency causing the exhausted phenotype. She was obsessed with it, and here we are. So, does encounter with metabolic stress contribute to the terminally differentiated state? And so what happens when T cells differentiate while their mitochondria are being inactivated, so to speak, by hypoxia? So here's what happens, right? If you take B16 melanoma and you put it into mice and then you infuse the mice with pimenidazole, and again, here's that same plot. If you look at the degree of hypoxia in these cells using this pimenidazole antibody, which this is just showing us that they're not very good. I want to make sure that we're very clear. The exhaustion field is a little hot button, right? But the key point here is that exhausted T cells still do something. They still make gamma interferon. They're highly lytic. They are proliferating. They just don't accumulate. But what they do lose is polyfunctionality, that ability to self-renew and the ability to make many cytokines. So we're just showing this here with IL-2 production. But here's the key point, is that when you look at their hypoxia staining, the PD-1 positive, the PD-1 high, TIM3 positive cells are uniformly experiencing hypoxia. All of them are severely hypoxic. And that's not the case with the other markers. Now, you can also just stain for HIF-1 alpha. It's not as dynamic as this pimenidazole antibody, but it's showing us the same thing. So exposure to hypoxia, we kind of reasoned, may be part of the way that these cells differentiate, because it was just so, it was a path these cells were on. So we went and broke it down. I'm going to leave out some of the intermediate parts, but we wanted to understand what was the experience of hypoxia in vitro doing to cells in isolation. And so we identified, we ruled out a couple things. We knew there was a rich literature about culturing cells in hypoxia, but that didn't take into consideration a main driver of the terminally differentiated phenotype, which is the continuous stimulation that occurs as T cells enter the tumor. So we asked a very simple question. If you have T cells experiencing hypoxia, but they're also getting other strong signals, what happens? And that's what we did. We generated a model that was based originally in a co-culture system with tumor cells. But for the interest of time, I'm going to talk to you about this model, where we activated T cells with off-the-shelf stimulatory beads overnight. And then we either removed the beads magnetically, or we kept the beads in. Just kept them in co-culture with these anti-CD3 and CD28 coated beads. But then we did that either at ambient normoxia, 20% O2, or in a hypoxia chamber that's sent to 1.5% O2. And we let that go, and then we remove the beads and we expand the cells in IL-2 and normoxia. Because we don't want to look at acute problems. We want to look at what happens after, even after we remove that stimulus. Okay, so this is a model system. It's a model. It's not perfect. But one of the things that we found was that if the T cells are experiencing continuous stimulation, or if they're experiencing hypoxia, you get these intermediate phenotypes, and this is just PD-1 and TIM3 expression. But the experience of continuous activation in the presence of hypoxia promoted a persistent upregulation of PD-1 and TIM3. So most of the cells had this kind of terminally differentiated phenotype. And I know most of you are thinking, well, that's just co-inhibitory molecules. It doesn't mean anything. So if you've been, read Nature recently, everybody's excited about this molecule, TOX. This is a transcription factor that's associated with terminal exhaustion, and we can find indeed that if cells are experiencing continuous activation under hypoxia, they also upregulate this transcription factor TOX. But the proof is really in the cytokine pudding, and if you re-stimulate those cells, what we find is this just kind of beautiful. I love the kind of dichotomy of these data. Continuous activation, T cells are totally fine. They're very good T cells. If T cells are cultured in hypoxia, consistent with previous literature, they're fantastic T cells. They do very, very well. But the experience of both simultaneously promoted a loss of polyfunctionality and a terminally differentiated phenotype from a functional perspective. So that was really exciting. So we had this idea that it wasn't just hypoxia, it was the experience of a lot of signaling in the presence of hypoxia. So what does that mean? What was actually happening? And so we want to check our metabolic boxes off. And so we can see loss of functional mitochondrial mass here with our MitoTracker dye. We can see repression of the PGC1 alpha molecule only when these cells are experiencing both, a little bit when the T cells are continuously activated. And if we look in the seahorse instruments, we can see the red line is the continuous plus hypoxia, is that these cells have more of a flat line and no respiratory capacity. So by all measurements, these cells looked exhausted-like. We're not going to call them exhausted, but they looked terminally differentiated. So exposure to hypoxia under conditions of continuous activation induced this persistent dysfunction. So what was actually happening? Now we had a system where we could decode the mechanism behind it, because it wasn't the dirty environment of a tumor, but rather we had these discrete inputs. And so one of the things that continuous activation upregulates, we know this from the literature, is a pattern of transcriptional repression that's driven, at least in part, by BLIMP1, a very well-known transcriptional repressor. And you can see terminally differentiated T cells, but even the PD-1 intermediates upregulates the expression of BLIMP1. What we found is our in vitro system really drove the expression of BLIMP1, this transcriptional repressor, such that the combination really had the highest expression of this. And so we reasoned that maybe BLIMP might repress some of our molecules that we like. And indeed, that's what we found, is that if you just take 293 T cells, for instance, and you overexpress BLIMP, you lose protein of PGC1 alpha. If you think this is a bad Western blot, you're right. But we did the real experiment, which is we used a PGC1 alpha promoter driving luciferase and showed that as you titrate in BLIMP, you lose the activity of the PGC1 alpha promoter. So we think that BLIMP is being turned on and that represses PGC1 alpha. Now, if you have BLIMP deficient T cells, those T cells are unable to be rendered dysfunctional in the continuous activation under hypoxia system. You can see this is double producers in wild type or BLIMP knockouts. But more importantly, BLIMP knockout T cells in vivo don't have this loss of functional mitochondrial mass, which was pretty exciting, and they don't repress the expression of PGC1 alpha. So we think that BLIMP is a big driver of this process. So that now led us to believe, what does PGC1 alpha do? What is it actually doing that changes how we respond to hypoxia? And so PGC1 alpha does a lot of things. We like it in the lab because it does a lot of things. It's not a master regulator. It's not like TOX or FOXP3 or something like that. It doesn't have any real effect on its own, but it collaborates with several different other important signaling pathways to orchestrate a whole degree of metabolic reprogramming. And in the lab, we've looked at mitochondrial biogenesis, we've looked at mitochondrial fusion, but the one I'm going to talk about today is antioxidant activity. The fact that when you make new mitochondria, you have to load them up with things that are going to mitigate all of the reactive oxygen species that are going to happen, that are going to be generated when the cells start doing oxphos. What we found is that we could stain for mitochondrial reactive oxygen species using this dye called MitoSox. And you could find that as T cells terminally differentiate in vivo, they upregulate, they accumulate a lot of mitochondrial reactive oxygen species, which you can see tabulated here. So that was an important thing to find. So the key question is, was that also happening in our in vitro system? And it absolutely was. When cells were experiencing continuous activation under hypoxia, they started to accumulate mitochondrial reactive oxygen species. So this was important. We wanted to understand a little bit more about what that reactive oxygen species was doing. So how can we do that under all of the things that we know hypoxia does? So it's seemingly counterintuitive that hypoxia would induce reactive oxygen species, because don't you need oxygen? But the fact is, hypoxia is not anoxia. There is still oxygen present. And as oxygen levels drop, you start to generate mitochondrial ROS at complex 1, this first step in the electron transport chain. So how could we assess that in isolation is that, and that's what this is delineating here, is that we could actually use an old drug called antimycin A, which inhibits complex 3 at this place. And when you do that, you mimic what hypoxia looks like without being hypoxic. And that was able to generate ROS. What was really cool, and I love these kinds of experiments, is that antimycin A alone was able to generate mitochondrial ROS. And this has been known before, that you can use this as a ROS generator. But what was really cool is you could add another inhibitor, rotenone, and bring it back down to baseline. So rather than just simply neutralizing the ROS, you can actually add a second inhibitor on top of the first inhibitor and go back and rescue the phenotype. So if you generate a lot of mitochondrial ROS, you generate exhausted T cells. They're terminally differentiated, PD-1 high, TIM3 positive, and they don't make cytokines very well. This is an overnight activation with these molecules, in case you guys don't believe me that we're just poisoning the cells. They aren't acutely toxic to the T cells, but it rather changes the way they differentiate over time. So it's not the loss. This is a complete crazy set of experiments, and it was years trying to get this stuff together, is that it's not the loss of mitochondria that's the problem, because when you inhibit with rotenone and antimycin A, you shut down the whole thing. But it's actually the presence of ROS that induces the dysfunctional state. So it's not the loss of functional mitochondria, it's the accumulation of dysfunctional mitochondria that really drives this process. So what was ROS actually doing? I know we're getting really deep into the rabbit hole here, but it's kind of neat. One of the things that ROS does a lot of things, so we wanted to drill down on what it might actually do. And so one of the things that has been reported, and we all know this because you can add peroxide to your cells to look at hyperphosphorylation. And that's exactly one of the things that ROS does, is it actually inhibits phosphatases. And so we thought that maybe intolerable levels of ROS might drive a kind of persistent phenotype, a persistent tyrosine phosphorylation. So we just took the old 4G10 molecule, that phosphotyrosine antibody that we used to use, and we just stained T cells with it. And kind of unsurprisingly, T cells that are terminally differentiated have high levels of tyrosine phosphorylation. I mean, they're being chronically activated in vivo, that's probably what's happening. And the same thing happens in our continuous activation. But what was really neat is that if you just do the antimycin A thing, just turn on ROS, you get hyperphosphorylation of all the phosphotyrosines in the cell, which was pretty neat. And so one of the phosphotyrosines, of course, that might be important is if you turn on a tyrosine phosphorylation cascade, you may get NFAT in the nucleus, which we know, of course, is bad unless it is with AP1, for instance. It can drive exhaustion on its own. And so kind of the idea behind all of this is that when you have these intolerable levels of ROS, you're kind of keeping NFAT in the nucleus all the time because you're inhibiting all your phosphatases. And if you want to do a kind of a wacky experiment, you can actually culture cells in sodium orthovanadate, a phosphatase inhibitor, and it's not very good for cells, but the cells it does leave behind have a terminally differentiated phenotype of being PD-1 high, TIM3 positive, and turning on BLIMP and TOX. So, I know it's a lot to see, but continuous stimulation represses the machinery needed to mitigate hypoxia-induced ROS, and that results in this kind of pathologically elevated phosphorylation cascade. So does this matter? So remember that RNA interference I told you about before? We knocked down the complex 1? What was really cool is the reason why we used RNA interference is because RNA interference is reversible. CRISPR, you can't come back from a deletion, right? So we were able to actually generate tumors that had a DOX-inducible RNA interference. So the tumors are totally wild type, and then we can render them less oxidative in vitro in here and in vivo. That 48 hours of going on doxycycline turns on this RNA interference, and it renders the tumors less hypoxic. And so, and here's the T cell hypoxia if we stain for p-manitazole by flow. And so what was really neat is that if you have the mouse and you don't give it dox, it's metabolically wild type, and PD-1 doesn't work. But if you then add dox and give anti-PD-1 and interrupt that hypoxia, you're then able to get a really striking synergy, and most of the mice clear their tumors with anti-PD-1. So this model of kind of metabolic exhaustion is that the persistent stimulation drives a pattern of transcriptional repression that changes the way that you deal with hypoxia. And so it's kind of a mix of two things, is that persistent stimulation changes the way you deal with hypoxia, and hypoxia feeds forward and changes the way that you experience other signals. And one of those mechanisms is by phosphatase inhibition. However, I'll tell you that there's a lot of other things that ROS does, and we're excited about epigenetic remodeling and lipid storage changes that happen in response to ROS. Okay, that's fun. So, sorry, that's like the second time I've given that story. So, our lab is really excited about metabolic changes with immunotherapy, and I think the cool part about it is that metabolism is so central to biology, and it's such a central defect in anti-tumor immunity that there's a lot of axes that you can change. So one of the things we're interested, of course, is in adoptive cell therapy, and how do you metabolically change adoptive cell therapies, like putting PGC1 alpha back into T cells, like into CAR T cells, for instance, which is something we're excited about. How do we change immune-excluded tumors, T cells that are there, but they're not in the tumor bed? We think hypoxia mitigation may be a great place to do this. We have a clinical trial of using metformin, which is a complex 1 inhibitor. It's an inhibitor of that molecule that we knock down in combination with anti-PD-1. We have a lot of excitement about using TNFR stimulators like 4-1BB and OX40 agonists. Those we have linked to mitochondrial reprogramming in the T cell. And in cases of immune desert tumors, I don't think we're even out of the woods yet, because we recently reported that you can actually stimulate the immune system with an oncolytic virus and then encode metabolic reprogramming in the virus itself, which is brand new. But all of these things are focused on the T cell, on CD8 T cells. We've been excited about CD8 T cells, for a while. They're cool. But with all of the focus, the fact of the matter is the majority of T cells in the tumor are not CD8 T cells. They're regulatory T cells. They're tolerogenic T cells. So this is where I did my postdoc studying regulatory T cells, and this is how my lab got started. But it took us a long time because FOXP3 reporters take forever to breed. And so Treg cells are these highly overrepresented T cells in the tumor. You can see that this is in the lymph node. They're a very small proportion of T cells, right? But in the tumor, the majority of T cells are regulatory. But they're not just idly standing there, right? They're actively proliferating, right? Most of them are Ki67 positive, and a good proportion of the Ki67 positive cells are in S phase, right? So this environment that I told you is so bad, so toxic, Treg cells are fine there. So why are Treg cells so fine there? This is something that I've really been struggling with since I started my lab. You can get rid of Tregs, but it's not good, right? Treg cells, if you get rid of Treg cells with a genetic system, the tumors melt away, but then the mouse dies from autoimmunity. So you can't just get rid of Tregs, you have to change them. And so we kind of hypothesized the Treg cells must have a metabolic profile that's distinct, that allows them to thrive in the tumor. And if we could identify what that metabolic profile was, we could target it, and then maybe only take care of Treg cells that are in the tumor. So how do we do this? We use the same glucose tracer that we had before called 2-NBDG. You can infuse it into mice, but these mice are FOXP3 reporter mice. And consistent with data that came from several groups, Treg cells, unlike their conventional counterparts, hate glucose. They won't take up the tracer even if you bathe them in the tracer. They really do not like taking up glucose. There's a subset that do, the small proportion, so we hypothesized maybe those are important. Now, what was really cool is that this tracer is a live cell tracer, and this is a reporter. So we could actually sort out cells based on their metabolism and then assess them functionally. And unlike CD8 T cells, which you can just look for cytokines, Treg cells, their suppressive activity, you have to measure indirectly. So what we do is you could sort out the Treg cells based on their metabolism, and then you just co-culture them. It's called a suppression assay. You co-culture them with a proliferation dye labeled conventional T cells. So if you don't have any Tregs, this is you can see the dye dilutes. And what was really exciting was that when Treg cells did take up glucose, that small proportion of them that did take up glucose, they were actually not very good Tregs. And it was actually these metabolically inactive, these less glucose-avid Tregs that were fantastic Treg cells. So What does that mean? So we took out those cells, and because, again, they were live, we could sort them out based on their metabolism, we did RNA sequencing on these samples. And what was exciting about this is that NBDG low Tregs, so the less glucose avid Tregs, they were fantastic Tregs, and that the high Tregs, the ones that took up this tracer, they weren't unstable. They didn't stop becoming Tregs. They were just a little bit less like Tregs. So all of these Treg signature genes were lower in glucose avid Treg cells, with the exception of a few notable exceptions. So we then went to the RNA sequencing and said, okay, so they look a little bit less like Tregs, but can we get a sense about what kind of metabolism they were doing? So we went to, we just looked at a bunch of metabolic genes. And because I'm running out of time and you guys don't want to hear me talk forever, I'm going to focus on these two genes. Glycolysis was interestingly all over the map, even though those cells didn't take up glucose. So all these genes, these are all the genes involved in glycolysis. But the NBDG low Treg cells upregulated two genes very significantly, LDH and this molecule, SLC16A1. All the SLC genes usually people throw away, but they're actually very important. What we found is that if you take Treg cells and you just culture them in low glucose conditions, they upregulate these things. This molecule, SLC16A1 and LDH, the terminal steps of glycolysis. And if you remember from those undergraduate biochemistry lectures that you probably forgot about, glycolysis is reversible, right? That glycolysis is not a one-way street. All of those steps are reversible with a few notable exceptions. So in fact, this molecule, which is called MCT1, or monocarboxylate transporter 1, transports monocarboxylates. And one of the most highly prevalent monocarboxylates in the tumor is lactic acid, the byproduct of all that fermentative glycolysis that I was alluding to. And in fact, in areas of the tumor, the lactate concentration can get as high as 50 millimolar, but if you just squeeze a tumor and measure the average lactate, it's about 10. So this is a really important thing. We think that glycolysis is a one-way street. But in fact, if you may remember things like the Cori cycle or the fact that your nervous system runs on lactates, is that most of the enzymatic reactions are reversible and they can interconvert. And so we hypothesized that lactic acid may be utilized as a fuel by Treg cells. And it occurs through this molecule MCT1 that forms a heterodimer with CD147. The only reason I bring that up is that before there was CD25, CD147 was thought to be the marker for Treg cells in humans. So we hope, we were excited about what this might mean. And then it can be converted and used. And so what was really exciting, we know lactic acid is an immunosuppressive molecule. If you take conventional T cells and you put them in high concentrations of lactic acid, they slow down. We've known that for a long time. But if you take Treg cells and put them in high concentrations of lactic acid, they tolerate it. In fact, they might even like it. And they're able to hang on to these very acidic conditions, and they don't lose their function by being in the presence of lactic acid. So being around lactic acid was important, but were they actually utilizing it? And we utilized an old school assay where we're able to measure the pH changes intracellularly that are induced by lactic acid. We could find that Treg cells uniquely are able to be acidified when you treat them with lactic acid. So they're able to take it up. And if you look at the cells that take up lactic acid, they look like great Tregs. They express CD44 and neuropilin. So we then had to do real metabolism, which was trace lactic acid. And to make a very long story short, what I will tell you is that we're able to show that lactic acid does get efficiently taken up by regulatory T cells and interconverted into TCA cycle intermediates, but it also is utilized in higher order intermediates. In other words, they're utilizing lactate and they're utilizing the carbons. They're not just burning them, they're utilizing the carbons to support their biology. So what are the requirements? What is the requirement for lactate uptake in vivo? So what we did is we obtained, in collaboration with Jeff Rothstein at Hopkins, a floxed allele for that MCT1 molecule. That molecule that we found was upregulated in Treg cells. If you delete that transporter, the cells can't take up lactate. But in the periphery, in the lymph nodes, everything is fine. The Treg cells, the mice don't get sick, and the Treg cells are able to suppress if we put them into a suppression assay. But if we then challenge the mice with a tumor, we have this very slow indolent tumor growth. So in that lactate-rich environment, the Treg cells get in and they stop working. The CD8 T cells in the tumor microenvironment proliferate more, the conventional cells do, and they're very, very good cytokine producers. They are double producers of TNF and gamma interferon. But the Treg cells themselves, they start destabilizing. They're unable to support that rapid proliferation we were talking about. And more importantly, when we put them into a suppression assay, they lose the ability to suppress conventional T cells, and they even start making gamma interferon. So this lactic acid, is not just a fuel source, but it's helping support the biology of Treg cells. All right, there's a lot of information, but I wanted you guys to be excited about metabolism. So the lab is really excited about lowering metabolic barriers to anti-tumor immunity. That could be bolstering the T cell, providing metabolic support to tumor-reactive T cells. That could be reprogramming the tumor, changing the way that it does metabolism to rebalance the environment. But we can't forget about these guys, that there are immunosuppressive relationships that are formed that utilize unappreciated metabolic pathways. With that, I'd like to thank the people in the lab that did the work. Plaid is our uniform in the lab. Nicole drove the hypoxia stuff, Mac is driving the acidosis stuff, and Ashley did the tumor cell metabolism stuff. And we couldn't do this without fantastic collaborations with Amanda Pahalik, who's a blimp biologist working at Pitt, and Jeff Rothstein for the animals. Happy to take any questions, and sorry if I went over.
I knew I was going to regret not paying attention to my metabolism course in medical school. I just noticed two things. So one was in the antimycin-treated cells, you had a picture where actin seemed to change its distribution, but you didn't mention anything about why you showed that. And the second question is, I also noted that EBI3 was particularly high in the Tregs that weren't uptaking glucose. And the question there was, do you see P35 as well?
Yeah, great question. So the first thing is the actin in that experiment, I think, was just to show you what the cytoplasm looked like versus the nucleus, to kind of show you the localization. However, we do know that, in all honesty, cytoskeletal rearrangement is extraordinarily metabolically demanding. And so one of the things we're studying in the lab are the metabolic pathways that lead to good motility, but still in its infancy. Your second question is I think right dead on. So P35 is difficult to detect. We've known, Dario's talked about this quite a bit. We do see a slight signature for P35 actually in the highly glycolytic Treg cells. So that's an interesting thing, actually, is that the glucose-avid Treg cells are still Tregs, but we think that they suppress by different mechanisms than the non-glucose-avid Tregs. But we're still the measuring IL-35 coming out of Treg cells is still tough to do. But we do have EBI3 reporters as well as P35 reporters now in our lab, so this is something we can explore.
Yeah, lactate in the tumor microenvironment can affect a couple different cell types. And so Oscar Colegio and Ruslan Metsutov's lab a couple years back had shown that tumor-associated myeloid cells or macrophages utilize lactate as well and uptake that. So any thoughts about that contribution potentially in the tumor microenvironment? Yeah, no, this is, I really want to talk about lactate here for that reason, because I think what we're finding is that many immunosuppressive cell types utilize these alternative sources of fuel. And lactate is just one of the things, actually, that that MCT1, it's a very promiscuous transporter. So it transports all kinds of things, including short-chain fats like butyrates and ketone bodies as well. So all of these, but these are all fuel sources that are very prevalent in the tissues. And so we think that this is a phenotype of being able to survive and suppress in the tissues. And I think that the story in tumor-associated macrophages, I think, is part and parcel of that. Would MCT1 as a potential pharmaceutical target be too likely to have toxicity elsewhere, do you think, or are you thinking about that at all?
No, actually, so there's several, so they were MCT1 inhibitors and antibodies have both been tried in the clinic in the days of just trying to starve tumor cells to inhibit glycolytic tumor cells. So many of these agents are now being repurposed in combination with immune-based therapies. We have utilized both a MCT1/2 selective inhibitor in combination with adoptive cell therapy and anti-PD-1 in mouse models. And we see a very nice synergy, but it's tough to drill down on what is actually a Treg phenotype versus changing the microenvironment as a whole.
That was a fantastic talk. What I'm wondering about is, how much of this, especially this switch to this, the exhaustion phenotype and the metabolic switch, how much of that do you think is due to need to survive, and you're taking away certain things away from those cells, versus actually like programming and driving the process?
Yeah, I think it's a fantastic question, but I think it's driven in part by kind of trying to reform how we think about the terminal exhaustion phenotype, right? That terminally differentiated phenotype is inherently a stressful pathologic response. And so I think many of these things are driven by the fact you suppress PGC1 alpha because you need to do something, right? And even though it's pathologically amplified, the fact is we know that this continuum exists for a reason, right? That you drive to terminal differentiation for a reason. And so what we're trying to explore now is what are the survival type phenotypes, because metabolic reprogramming is of course important for all cells, not just T cells, but then being able to assess what are the individual pathways that contribute to that self-sufficiency or lack of self-sufficiency, and is that just what happens as you differentiate, you become more dependent on your environment. So I think it's a multifactorial question. We're very excited about what those things might mean, but don't know all the answers yet. Great, thanks very much.